Nothing but net, and I promise that was my first shot. Hey Bio30s, I'm here to chat about our next unit actually, which is going to be cells and DNA. So this is going to be 2 chapters. It's going to be chapter 17 and 20. So we're going to do a little bit of chapter 17 the next couple of weeks, move on to chapter 20, talk about DNA, and then we'll move forward afterwards with some genetics. So starting off here, 17.1, I'd like to discuss the cell cycle, so the stages that a cell goes through, as well as the process of mitosis. So that's kind of where we're heading today. So the cell cycle, basically 90% of the cell is actually, 90% of the cell cycle is actually in a phase called interphase. This actually is no cell division whatsoever. It's basically the cell doing its daily activities, whether that be growing, repairing. If you're a heart cell, you're doing your thing. If you're a skin cell, you're doing your thing. If you're a white blood cell, you're doing your thing. Copying and replicating DNA, this all occurs during interphase. There's actually no division happening during interphase. Interphase can then be further subdivided, and I'll highlight these right here. So we can subdivide them into gap 1. That doesn't look very good. Let's try this. White. So gap 1, S phase, gap 2. So during gap 1 or G1, we basically have cell growth and the cell is doing its regular functioning. So just going about its daily cell life. Then we reach S phase, and when I think of S phase, I like to think of synthesis, S for synthesis. This means DNA is going to be duplicated. This means our single-stranded chromosomes are going to become double-stranded chromosomes, and I'll explain what that means in a second. Then we have gap 2, which is similar to gap 1, but now the cell is doing its cellular activity, yes, but it's also preparing for the process of cell division, also known as mitosis. So here's a look at what cell cycle looks like. You'll notice that once again, 90% of cell cycle, right? 90% of the cycle is actually interphase, going from G1 into S phase, then G2, then mitosis, and then back to G1. So it's a cycle, and it doesn't matter where you start in this cycle as long as you get the order correct. So G1 to S phase to G2, and then further on to mitosis. So let's talk about chromosomes, and best way I can explain chromosomes is using these markers. So let's chat about chromosomes a little bit. Remember, chromosomes are packets or bundles of DNA, and they contain genes. These give you the traits that give you eye color, hair color, your height, things like that. So chromosomes can be uncondensed or condensed. Uncondensed means they're long and kind of stringy. They're very long, thin, very, very hard to see under a microscope. So when chromosomes are packed in the nucleus, they're uncondensed. They're long, kind of stringy, thin chromosomes. When the nucleus disappears during mitosis, they become condensed. So they actually get shorter, they get thicker, and they're actually able to be seen under a microscope. Now, when we talk about chromosomes, these 2 images here, this is a chromosome and this is a chromosome. So this is a chromosome and this is a chromosome, right? They both would be considered one chromosome. However, one is unduplicated and one is duplicated. When we have a duplicated chromosome, we call it sister chromatids. So chromatid, chromatid. 2 chromatids put together makes a chromosome. But this is one unduplicated, this is one duplicated. Now, here's the big thing to remember. This and this are pretty much the same thing because all the information here is exactly the same as the information here. There's no new information, right? So in terms of the information present, anything you see here is going to be here and here. Meaning, a duplicated or double-stranded chromosome, right, still counts as one chromosome. It's not 2. 2 chromatids, but 1 full chromosome. One final thing to note here, that the chromatids are joined in the center by what's called a centromere. So a centromere would be centromere would be right here. holding the two chromatids together. Okay, now that we understand chromatids, chromosomes, and interphase, let's get moving on to mitosis. So, during mitosis, each daughter cell gets one single-stranded chromosome. This is why we have to duplicate them, right? One over here and one over here. Now, cells will use a single strand of DNA to make a complementary strand. This is called DNA replication, which we'll talk about in chapter 20. All you need to know right now is DNA replication occurs during S phase, which once again is the synthesis phase of DNA during interphase. A somatic cell, also known as a body cell, contains 46 chromosomes or 46 packets of DNA. So every human cell in the body that's a body cell is 46, with the exception, of course, of sex cells. They contain 23. So any body cell in the body right now, your skin cell, your heart cell, your muscle cell, they're all undergoing mitosis, okay? Mitosis, once again, 10% of the cell cycle. It occurs in all somatic body cells, and we're going to start off here with prophase. So here's what's happening in prophase. During prophase, the nucleus is breaking down, that nuclear membrane is disappearing. The chromosomes are going from the long condensed version, the long uncondensed version, and now they're getting shorter and they're actually getting condensed. In animal cells, centrioles will appear and start moving to opposite poles. So one will go to the top to the north pole and one will go to the bottom to the south pole. And we also get spindle fibers as well. So we start to get these long threads starting to form within the cell. This is prophase. Sometimes prophase can be broken down to early and late prophase. So early prophase, nuclear membrane breaking down, chromosomes condensing. And then late prophase, spindle fibers attaching to the chromosomes as well as the centrioles. Think of centrioles like anchors. We can see an anchor, and then spindle fibers attaching to those anchors. Okay? Next, we're going to have metaphase, and I'll just grab a couple more markers to show you metaphase here. So in metaphase, the chromosomes will line up in the middle of the cell, the equatorial plate. Remember, the chromosomes at this point have gone through gap 1, S phase, so they duplicated their DNA, so now all chromosomes are double-stranded. You have 46 double-stranded chromosomes, okay? Those sister chromatids are now going to line up. So there's going to be one here, one here, one here, one here, a big, long kind of mass of chromosomes in the center. Now, they're not all neatly spread out evenly. Usually it appears as a dark mass under a microscope, but this is generally the best phase of mitosis to look at chromosomes for karyotyping, which we'll discuss at the end of chapter 17. That's kind of like gene mapping and figuring out what's in your chromosomes or what traits you get. Anyway, so the chromosomes will all line up in the middle of the cell. They are attached to the centrioles by the spindle fibers, and it also attaches to the centromere. I know those words are very similar. Often they get confused. Okay, anaphase is now when we start pulling the chromosomes apart. So now we're going to pull. So one chromatid goes this way, one chromatid goes this way, and now what's essentially happening is you're splitting the chromatids, the sister chromatids, meaning now 46 single-stranded chromosomes go this way, 46 single-stranded chromosomes go the other way. Now you're probably wondering, but wait, what about wouldn't one cell be missing out on traits or genes? No, they wouldn't, because remember, these are exactly the same DNA on either one. So one cell gets one, the other cell gets one. That's anaphase. So now we're pulling them apart. Finally, we're going to end with telophase, which is where we now have two new nuclei forming. The chromosomes start to uncondense, the nuclear membrane reforms, and we get the cytoplasm starting to pinch off. We get a little bit of pinching action of the cytoplasm to start basically the splitting process. And the ultimate splitting process will then end with the process of cytokinesis, the splitting of the cytoplasm. So now the cytoplasm will completely split up, where we get two new daughter cells. Each daughter cell is identical to the parent cell, both 46 chromosomes, and they're all going to be 46 single-stranded. So one cell over here, 46 single-stranded, one cell over here, 46 single-stranded. Now, that's the process of mitosis. Once again, ending with cytokinesis. Organelles will be evenly distributed, so some mitochondria here, mitochondria there. We get some ribosomes here, ribosomes there. We'll evenly distribute the organelles, and now we have 2 brand new cells. Away we go. So, just a recap of everything before we end. Interphase, this is where the cell is just going about its daily business. Gap 1, S phase, and gap 2, cellular function, copying DNA, building and repairing, and then we get into mitosis, where we have prophase, the nuclear membrane. Notice how the chromosomes are going from thin, chromatin, right? Thin, uncondensed chromosomes, to thicker, shorter, condensed chromosomes at the end of prophase. Spindle fibers are forming, and centrioles are moving to the poles. Next, metaphase, lining up in the middle. Anaphase, pulling apart, remember, pulling apart chromatids. This is very important to remember, especially when we talk about the other type of cell division, which is meiosis. Finally, telophase cytokinesis, we reform 2 new nuclei, each with 46 chromosomes. The cytoplasm pinches off, and we have ourselves the end of mitosis and cell division. So that's all I have for you today. Next lesson, we're going to cover meiosis, so the other type of cell division, how you make sex cells and gametes. So thanks for watching, and we'll chat soon.